Usually I'm a professor of physics and biology in Emory University in Atlanta, but today I'm standing here on behalf of the Aspen Center for Physics, and I welcome you all to this six Heinz Pagels lectures of the 2016. Tonight's speaker is Leonid Mirny, professor from MIT, however, before I turn the floor to him, I will say a few words about the names I've just mentioned, the Aspen Center, the Pagels lectures, and Leonid himself. So first, the Aspen Center. So what is this center? Every summer, about 500 leading scientists from across the world assemble here at the center for a few weeks at a time, but I'm here for 2 weeks this year. And the center is an iconic institution in physics. The impact of the center is by far a lot larger than the, let's say, the budget of the center is. The center is an incubator. It's a place where we brainstorm, where we collaborate, where we meet other scientists. Some of these scientists serendipitously become our friends and our scientific collaborators later on, and we don't know this when we come here. We just talk to the people, and it just happens. So personally, I've been coming to the ACP, to the Aspen Center for Physics, for 12 years now, and every visit here has been magical. It results in new collaborations, a new understanding of things that I thought I already understood, but it turns out I didn't, and results in new ideas, new friendships, that last for decades. Some of my most impactful work, for example, wouldn't exist if not for this center. So why is ACP so impactful? Well, I don't know, but maybe it's a cool mountain air. It could be, right? It could also be that it's Colorado scenery, which is quite inspiring. Maybe it's the scientific climate here at the center, which sort of shelters us, the professors at various universities, from distractions of everyday life. And it's built around opportunities for sort of fresh new activities, for in-depth discussions, unstructured but nonetheless very in-depth discussions, which are intensely focused, right? We spend here 2 or 3 weeks at a time, and we spend here since 8 in the morning till midnight just talking to people and trying to solve the problems. We come here with questions, and these questions often get solved, and then we leave the center with tens of other questions which we now need to solve for the rest of the year. And so what can be more exciting than that, where a sort of 2-week stay at the center creates this new idea that are going to last you for a couple of years into the future. And so what can be more impactful than that, right? So we're very thankful to the National Science Foundation, which has been funding this center in part as the investment in the scientific infrastructure of the country, and we hope that this is going to continue for the years to come, and the center is going to serve the country in this capacity for many years to come. So the second thing that I have to talk about is the name of this lecture. So this lecture series that you are attending are named after Heinz Pagels. He was a long-term participant here at the Aspen Center for Physics at ACP. He was a professor at Rockefeller University at New York City and a leading figure in particle physics. He was a distinguished educator and author of many popular books, and he was the president of the International League for Human Rights. Another interesting thing about him is that his work on chaos theory was an inspiration for a character of Ian Malcolm in the Jurassic Park book and the movies. So he died tragically here in Aspen when he was trying to climb the Pyramid Peak in 1988, and the friends here at ACP has decided that it would be a fitting tribute to his life if we name the lectures, the public lectures here at the center, in his honor. So the last thing I have to say is the introduction of our speaker today, but before I do that, let me remind you that the next event in this series, the 7th lecture, is going to be next week, next Thursday on August 25th. It's going to be given by Alexandra Walczak from École Normale Supérieure, and she's going to be talking about the physicist's view of immune system. So if you want to figure out, why it is that you get sick and then why don't you die because of that, then you should probably come next week. If you want to be on the mailing list for future events, there is a sign-up sheet right there at the entrance to the auditorium, and you should sign your names there. So today's speaker is Leonid Mirny. He's professor of physics and health science and technology at MIT, Massachusetts Institute of Technology. He has received his Master of Science degree from Weizmann Institute of Science in Israel in 1994, and then PhD from Harvard in 1998. He later was a junior fellow at Harvard University, and then in 2001 he moved a mile down the road to become a professor at Massachusetts Institute of Technology at MIT, and he's been there ever since. He is a fellow of the American Physical Society, and he has made his own contributions to our theoretical understanding of proteins, of physical structure of DNA, which is what he's going to, of how DNA is packed into the cells, which is what he's going to be talking about today, and how proteins in cells control which genes are going to be producing any future proteins. Interesting fact about Leonid is that he's also a stand-up comedian. For many years, he has been a captain of the Harvard University stand-up comedy team. This is a traditional team comedy game which is very popular among Russian immigrants to the United States. And in fact, Leonid and I have been friends with this comedy club for about 20 years, way before we became friends as scientists. So this evening, he will tell us about how about 2 meters of the human DNA can be packed into a micron-sized nucleus inside of every one of our cells. And there will be plenty of time for questions during and after the talk. He'll tell you when and how he's going to take the questions. And with that, let's welcome Leonid.
All right, so it's a great honor to speak here, to give a public lecture. Again, thank you for inviting me. As you realized, Ilya was also a stand-up comedian. All right, I'm not sure I can stand to the high standards of this profession, so I'll pretend to be a physicist now. So human genome, now in 3D. So let me start with something very familiar, with the DNA double helix that, of course, you've all seen. So what's important is that sort of in the structure, these elements in the staircase are bases. So and there are 4 types of bases, A, T, C, and G, and this is usually shown like this. So A's are complementary to T's, and G's are complementary to C's. So what's important is that the structure that is in every textbook, and is practically an iconic image, consists of 12 base pairs of DNA. Human genome contains 3 billion letters. So what we would like to do is to get a structure of 3 billion letters. So that's the statement of the problem. So if you were to ask biologists, most of them would tell you that most important technological advancement in biology in the last 30 years was our ability to read DNA. So in 2001, the first draft of the human genome was published. So we read 3 billion letters, approximately 3 billion letters of DNA, and that's what we got. It's certainly a lot of information. So, for example, a Wellcome collection in London, one of my favorite small museums in London, contains this beautiful stand with the human genome printed in font size 5. That's just one stand, so all 23 chromosomes and then X and Y chromosomes. You can browse the book. It's a very interesting reading. The amount of information, actually the number of letters, is approximately as many as in Encyclopedia Britannica. So what do we know about these letters? So first of all, it's a one-dimensional text, so it's just like a text, literally read line by line. So some of these regions are genes. Genes, however, constitute only less than 2% of the total genome. The rest is called, biologists call this junk DNA. Many are offended by this. So I would kind of follow the example of physicists. I would call this dark DNA. So let's call 98% of this dark DNA. And there are genes, obviously. And we certainly know much more about genes than about the dark DNA. So let me again give you a sort of molecular biology class, like molecular biology 101 in 3 minutes. So this is human genome. It's linear. It consists of all these letters. So it's made of DNA. So there are genes. As I told you, only less than 2% is genes. It's split into 23 chromosomes. So each chromosome is again a piece of DNA, but sort of the whole 3 billion letters are split into 23 chromosomes. So this is chromosome 1, chromosome 2, and so on. The total length of DNA in every single cell is about 6 feet. So that's a lot. That's especially a lot compared to the size of the cell. So the question is, how can 6 feet of DNA be packed inside a cell, or specifically inside a nucleus of a cell, which is only about 10 microns in diameter? So if this were DNA, If this were the length of DNA, the size of the nucleus would be much smaller than 1 pixel on my screen. And that's a pretty good screen. So it's much smaller than that, just the relative size. Now, if a nucleus, cell nucleus, was the size of a suitcase, then the DNA that's packed inside the suitcase should be approximately as long as the road from Aspen to Denver. Now, my daughter, when I kind of was practicing this conversation with my 13-year-old daughter, so she asked me, so what's the total length of DNA in all your cells? And I said, I never thought about it. So we have a lot of cells. So if you actually multiply 2 meters by the number of cells we have, it turns out that DNA from all of your cells can stretch from sun to earth and back 200 times. So you have a hell lot of DNA. It's a very, very long molecule, and it's packed inside relatively small volumes. Moreover, it's packed in a non-random way. So that's the question. How is human genome of 23 chromosomes folded in 3D, and how is it folded to allow genome function? And there are many functions of the genome. I'll just mention a few. So what do we know about this? Chromosomes were first visualized by Walter Fleming in 1882, and the name chromosomes means that it's visible in the microscope. It's something that's visible in the microscope without any additional staining. So, and he characterized chromosomes through the cell cycle and in different types of organisms. So, just 120 years later, we get somewhat high-resolution images where individual chromosomes are colored differently. They're stained in different colors. Again, you should realize that sort of chromosomes during most of the life of the cell are unpacked. So these chromosomes kind of are open, not very organized. When a cell needs to divide, it needs to pack its chromosomes, so chromosomes become very, very condensed and well organized. And in fact, you have 2 copies of each chromosome because you need to give this pack of chromosomes to each of the 2 copies when the cell is dividing. So that's the picture that I'll carry through the talk. This is interphase. So the most of the times the cell spends in this state when chromosomes are open, and when the cell needs to divide, it packs its chromosomes. So what else do we know? We know that chromosomes are affected in cancer. That's, again, from the works of Theodore Boveri, again, end of the 19th century. We know that chromosomes are highly altered in cancer. Here, for example, chromosome 1, we have only one pair of chromosomes. In this breast cancer cell line, we have 4 of these chromosomes, and they carry little pieces of chromosome 2. Chromosome 2, in turn, actually chromosome 3, yeah. Chromosome 2, in turn, carries a little piece of other chromosome. So chromosomes are messed up. So it's really, so the total, you have a different number of chromosomes, and pieces of chromosomes are fused to places where they're not supposed to be. We don't quite know whether it's a consequence of cancer or the cause of cancer. It's a discussion that's ongoing since the end of the 19th century, and still there is no clear answer. It's certainly something that's highly associated with cancer. So what else do we know? We know that genes in the genome are they're not passive elements. They're being read from time to time, and when a gene is being read, we say that the gene is active. And it means that the protein is being produced following the instructions written in the gene. So some genes are on, they're active, and some genes are off. And that's very important, again, for development, because all cells in our body have the same DNA. My retina and my liver has the same DNA, but they have totally different functions. And this is controlled by which genes are expressed in the liver. There are special genes expressed, and in the retina, different genes are expressed. So what controls which genes are expressed? So there are switches. I will just call them genomic switches. So there are switches that control activity of the genes. Again, I'm talking about the state of the cell when DNA is unpacked. When it's packed, all the switches are off. And then when chromosomes open up, all the switches again go into their right positions. Now, what's important about the switches is that they control genes, not those that are close to on the genome, but those that are close to in space. So for this switch, for example, there are 5 genes that are nearby because the chromosome is folded in this particular way. That's just a cartoon. And so these three genes are going to be on because they're almost touching the switch. And the other two genes are going to be off because they're somewhere further from the switch. So 3D folding is important because it controls which genes are active and which are not. So 3D folding is not just a question of how it beautifully looks in 3D, but it in fact answers the question of which genes are going to be activated and which are not. So there is a new technique, actually several new techniques, that allow us to examine chromosome folding inside cells. Again, the first structure of DNA double helix was solved by X-ray crystallography. It was purified, isolated, crystallized. It's completely in vitro system. So what I'm going to show you are actually data collected inside cells. So the method is called chromosome conformation capture. And thinking about it, I realized it's a paparazzi technology. It's kind of a random paparazzi technology. A random paparazzi would be just taking pictures of people on the street and then later identify who won the shot. So Hi-C is a technology, so chromosome conformation capture. I will call it Hi-C, that's the last. Again, it used to be called 3C for 3Cs, and then there was modification of it called 4C, and then 5C, and then people got tired of this and called everything else Hi-C. So this is Hi-C technology. What it does, it's essentially a chemical microscope. It captures all pieces of DNA that happen to be close in space. Then it reads DNA of these pieces and identifies where these regions came from in the genome. And in a single experiment, it does it billions of times. Again, it's allowed by the technology to read DNA cheaply and rapidly. So what do we get from this technology? Again, the technology was developed by Job Decker, our close collaborator at UMass Medical School in Worcester. And what you get these beautiful maps. And these maps are telling you how frequently different regions of the genome interact with other regions of the genome. So it's a map. Here you have, for example, one chromosome, and the darker the region is, the more frequently these regions interact. For example, if you look at zoom into one area, you see this dark diagonal. A dark diagonal tells you that there are lots of interactions between genomic neighbors. Regions that are close to each other in the genome, they interact a lot. So there are lots of interactions among neighbors. That's good. What else do we know? We see some interactions further from the diagonal. So these are interactions, rare interactions, so they're not as bright as what you see close to the diagonal. So these are rare interactions among distant regions of the genome. Two ends of the same chromosome, between chromosomes and so on. So these are interactions, they might be random, they may not be random, they might be good, they might be strange. But these are interactions between things that are not, between non-neighbors. What else do we know? We recently, as of a couple of years ago, we realized, we as a community, is that there are also fences in the genome. So there are some regions, and they, again, on these maps, they look like corners of the squares. So there are some regions of the genome that essentially work as fences. They prevent interactions across fences, and they allow lots of interactions within regions that are fenced on both sides. That's a very interesting organization that we did not know about 5 years ago. Everything I'm telling you is very recent. So the technique was developed in 2009. This data are from 2012. The rest will be, some of the papers that I'll talk about have been published in the last few months. So it's really a thrill for me to tell you about this. It's not something that's written in textbooks. So why is this important? Why this organization and sort of this interactions among neighbors is important? To some extent, it's important, again, because it controls which genes are regulated by which switches. So there are, so if we go back, in this example, this switch is on, but only this gene is on, and this one is off. This one is off, again, neighboring ones are off. This one is on, again, this gene is on, but this one is off. So why is this? So, but if you map fences on top of this picture, you see that fences control the range of activity of the switches. So this switch is off, so everything in this neighborhood is going to be off. This one is on, so everything, or many genes in this neighborhood are going to be on. That's a paradigm that emerges. It's not still a proven fact, but that's something that's started to emerge in the last year and a half, really. This picture and the role of these fences that we did not know about as just a couple of years ago. So how does it all work in 3D? Again, this tells you what we see in this data, but it doesn't really give you a comprehensive 3D picture. So what do we want to do as physicists? Again, as a physicist, I would like to get this beautiful high-C data, and not only make a poster for my wall, but also try to get some sort of 3D structures of the genome that would correspond to this data. And that's a very hard problem, because given the structures, I can compute this map, but it's really hard to go this way, from the map to the model. And it's not that different from the situation in science in the early 1950s, when the first X-ray data have been obtained by Rosalind Franklin, for DNA by Rosalind Franklin in Maurice Wilkins' lab, and then Watson and Crick tried to build a model, and so several labs tried to build a model of DNA that would be consistent with this data. And essentially it was trial and error approach. They tried different models and then see which ones would explain some of the features of the data. And we practically follow the same logic. We're going to try several models and see which models are consistent with the data. And again, as I told you, they did it for 12 bases. We want to do it for 3 billion bases. This data are already available for a range of organisms. Although it's a very new technology, we have these maps for the 3 billion bases of the human genome, for mouse, flies, some strains of yeast, and some species of bacteria. So how does it all look like? So in the first study that we published a couple of years ago, what we did, we decided to look at chromosome structures through the cell cycle. So as I told you, during most of the cell cycle, chromosomes are open, and there is some structure, there is some neighborhoods. Chromosomes are unpacked, and then the cell goes into a state where it packs all the chromosomes. So chromosomes should get packed, and the structure should change, and that's what we saw. This footprint of the structure is totally different when the chromosomes are packed. And then 2 hours later, it unpacks the chromosomes, and the structure again goes back to what it was just in the previous cell cycle. It's really magic how it happens. So what we decided to do first is to look at the structure of chromosomes in this packed state. Again, this is an iconic image of a chromosome. Essentially, these are 2 chromosomes for 2 daughter cells. So one will go to one daughter cell, another one will go to another daughter cell. They're tightly packed. And how DNA goes inside this chromosome, that's what we're trying to uncover. Let me maybe pause here for a second, sort of, if there are any questions at this point. We're kind of halfway through. Yes.
So often in science, you say always or never. It's like there's always exceptions. So when you start out earlier and said that when cells split, they're all turned off.
Yes, so that's okay.
Is that true? I was thinking of that.
It's a great question. At this point, as of 2015, everybody would say yes, all the genes are off during cell division. Right now, we start seeing that maybe a few genes escape this. But the paradigm, I would say, if you ask 10 biologists, 9.5 of them will tell you that yes, all the genes are off. That's one of the few things that seem to be well established. There might be some exceptions, but looks like these are kind of exotic cases. So genes are off during cell division. It doesn't mean that all the information about the switches, that all the switches got turned off. There may still be some, this is called pioneering factors. There might be some sort of additional marks that are still left on the genome, such that when it gets unpacked, it can rapidly put switches in the right positions. But genes are not active at this stage. Yes?
In your last slide, you talked about the packing and the unpacking, and maybe 2 slides ago.
Yes. Yeah.
Made it sound like it would happen in 2 hours. How frequently does that cycle happen? And
about once a day, roughly. So cells in a dish divide once a day, once in 2 days. Yeah. So this is very early stage. So just unpack chromosomes and everything start to look normal, really fast. Yes. Yes.
The life cycle of a cell is only 1 day in your body. Every cell divides every day?
Not every cell. So only stem cells do this frequent division. So cells that produce blood, epithelial cells of the skin divide not that frequently. Epithelial cells of the intestines divide very frequently. Your hair is growing. So few organs contain lots of stem cells, but other cells don't divide. Like neurons don't divide practically never, very rarely. Other cells divide very, very rarely. But so this was done on cells in a dish. And then we repeated this on fibroblasts taken from the skin. So they also divide quite fast. Yeah, let me take the last question.
Excuse me, and is that constant as a person ages, or does that slow down as they age?
It does slow down, sort of. Obviously, when a body is developing, cells divide quite actively. So in adults, the audience is mostly adults, very few infants. So cells, obviously, very few cells continue this fast division. Kind of a prototypical example is cells that are needed to produce blood cells, hematopoietic system. Okay, let's continue with this. So we try to solve the structure of the mitotic chromosome. So these packed chromosomes. We have high C data for this. So that was this beautiful thing that just linked on the screen. But we need an idea about the model. We cannot just start from the data. So you can open a textbook. Actually, this is an AP biology textbook from my son, who is sitting there in the front row. So when he was in high school, so I was actually working on this, and so I asked him for his textbook. So that's a textbook model. So this is DNA. It's wrapped around some protein cores, then forms this interesting structure, kind of high-order spiral. Then the spiral forms a series of rosettes, and then these rosettes go into the chromosome. So it's in every kind of molecular biology or AP biology textbook. This is Natural History Museum in London. The model is different. So it's DNA, it forms a helix, and then it forms a high-order helix, and another helix, and this helix goes in. They certainly love helices. Wikipedia, this must be true. DNA, some sort of zigzag pattern of proteins, kind of interesting fiber, loops, and then this loops form a helix, and this goes inside the chromosome. Lots of opportunity to test all these models against the data, because all these models were proposed based on some circumstantial data, and now we actually have direct measurements. So we try these models. What do I mean by we tried? So we take, for example, a Wikipedia-style model, and we actually implement it in computer simulations. We build a polymer, a long chain of DNA, where every region could, essentially, this would be essentially DNA, if you wish. And then we fold it according to this recipe. We let it kind of equilibrate thermally, kind of let it jiggle and wiggle, and then compute a simulated high-C map and compare it to the real high-C map, and we see no agreement whatsoever. So this model is dead. So then we did the same for the textbook model with the same result. For models similar, like multi-level spirals, the same result. Practically all the models in the popular, kind of, even in textbooks, turned out to be inconsistent with the data. I wouldn't say it was wrong, because it's a strong word, but they were not consistent with the data. So then we were kind of disappointed, because we were looking for a model that works. We were not on a crusade to kill all the models in textbooks. So then we started looking into the older literature, and it turned out that there were beautiful ideas in 1970s and 1980s, where people isolated chromosomes, spread them on a piece of glass, and did microscopy imaging. So the main idea of these models was that there should be loops. Loops and no other levels of organization. No helices and loops of loops and no rosettes practically, but essentially loops that emanate from the central scaffold. And we said, okay, let's try to build a model this way. So we would build a central scaffold, and then loops emanating from the scaffold. And then we tried different sizes of loops, and for a particular range of sizes, we got really, really good agreement with the data. And it turned out to be exactly the same size that some people measured, again, in some of the papers in the 80s. So that was a big surprise for us, that this particular class of models worked, and so no other class seemed to give an agreement. Again, it doesn't mean that there is no other model that would work, but at least we found one model out of many that didn't work, only this one did work. So how does it look like? So I will just rotate this model. So there are lots of loops inside a single chromosome. I will emphasize a few in color, such that you can see that they're highly disordered. So our idea about these chromosomes being very neatly packed is kind of silly. So it's very dense, but every single region is still crumpled to an extent. And this model gives very good agreement with the data. So that's our model of human chromosome during cell division. So still we have a question, so how does it get into this state? It's a nice model, it's interesting, but it should get there somehow. So we started again thinking and trying different models, and then we started collaborating with John Marco, a physicist from Northwestern University, who proposed an interesting idea. He said, what if DNA is compacted by this interesting mechanism that nobody have ever seen, but let's hypothesize a mechanism. We're physicists. So let's start with a hypothesis. Let's start with theory and see whether this theory works or not. So let's have, he calls this loop extrusion. Let's say this is a machinery, a motor, and it lands on a piece of DNA and it starts creating a loop. And it creates a larger and larger loop. And let me demonstrate this to you. So I brought some demonstration material. Okay, so let's say that's a piece of DNA. Essentially what the loop extrusion model suggests is that, so both of my hands are going to be a single motor. So it lands on a piece of DNA and it starts making this loop, and the loop gets larger and larger and larger. Say, well, you can't really fold much doing this, right? I was very skeptical, frankly. I was like, yeah, you can't really compactify things. You just create loops. Then I said, well, what if this one does the loop, and then so maybe you can help me, and so you're going to make another loop for me. So if you essentially land, you do this, and you can do the next one. So we have 3 loop extruding machines, and you keep doing this until your hands actually meet with the hands of your neighbor. You see, so you get a system of loops, yes. Yeah, at the end, you get a system, okay, you keep holding, I'll show this to the audience. You get a system of loops. That's exactly what we needed in our model. We needed a system of loops with a central scaffold, and their hands essentially form, if you raise your hands, thank you. And their hands are essentially forming the central scaffold of the chromosome and the system of loops. So, wow, that's interesting, maybe, thank you. It's actually first demonstration of this type of idea on the members of the audience. I usually need approval for experiments done on human subjects. I don't tell anybody. Okay, can this really work? So if I make this, if I simulate activity of these loop extruding machines, can it really fold chromosomes the way we know they should be folded in this state? So that would be our simulations of me. So essentially this would be like, so this is a jiggling piece of DNA, and this is this machinery that creates a larger and larger black loop. So there are 2 problems that this machine that this kind of mechanism should solve. The first problem is that you start with a DNA which is just a coil of mass. Remember, this is DNA during interphase when genes are active, so it's not particularly organized. And if I just compactify it, I would get something that's small, but it wouldn't really get this extended, elongated shape that the chromosome needs to have. The second problem is that each chromosome, as I told you, and again, I will continue my demonstration. So each chromosome gets duplicated during cell cycle. So because now I need to give these 2 chromosomes to 2 daughter cells. But when they're duplicated, they essentially run parallel to each other. So they run parallel to each other, and they're not particularly organized in any fashion before they get compactified. And now I need to compactify them somehow that red ones are separate from the blue ones. The other challenge is that machines that do this are the size much smaller than the size of the chromosome. So imagine like little insects trying to pull this chromosome apart and not really being able to say blue from red. How can it work? So that's the second problem. So let me show you what loop extrusion can do for the first problem. Just so it's a single chain, so I forget about 2 chromosomes for a second. It's a single chain. These red dots are actually these machines that will simulate, that can extrude loops. These are computer simulations. So it will first give you an overview. So you see the same model on the left and on the right. On the right, it's what we call an X-ray. So you don't see a chromosome, you don't see DNA chain in color, you see it in gray, but you can see these loop extruders. And they do the same job as our members of the audience did with this piece of string. So what you start seeing is that you get the scaffold, get the laser pointing. You get the scaffold of the chromosome being formed, and the chromosome gets, in fact, elongated. It gets the shape that it's supposed to get when when the chromosome is folded. So it's not just a kind of a spherical jumble of DNA, but it's an extended structure that resembles a real chromosome. And that was a big surprise. So you can condense things into extended structures with the scaffold and a system of loops. That was the main result. We need a scaffold and a system of loops. And then we say, okay, that's great. Let's try the second problem. Now we have two sister chromatids intertwined, and I will essentially let these loop extruders do their job. I'm not going to do anything else in the simulation other than let loop extruders create these little loops. And what you will start seeing is that each chromosome will become gradually bulkier because of the loops. It will become shorter, and it will become stiffer also. And keep watching. I need music for this. If you have some suggestions, come to me. That may work. Zarathustra. Yes. Zarathustra would do, yes. Great. That's right. Yes, so what you see at the end is that you automatically get two chromosomes, each condensed, and they're segregated from each other. So the only thing that it needs, in addition to these machines that create loops, it needs another machinery that is well known. It allows two strands of DNA to pass through each other. Otherwise, you would get things somewhat knotted. So these enzymes are called topoisomerase, and it allows two strands to pass. It cuts and it connects again. So there is such machine. It's known for already 30 years that this machine exists. They're absolutely essential. But they're not enough to condense. And these things, this loop extruders seem to be enough to condense, again, in theory. Okay, so what did we learn from this? First, we learned that to pack DNA from the interface to mitosis, to cell division, you may just need one mechanism, and it's possible that this function is performed by a particular class of proteins called structural maintenance of chromosomes. So it's known that these proteins are known to have a very important role in chromosome folding. These proteins are one of the best conserved proteins in evolution, all the way from archaean bacteria to human. Very few proteins are actually conserved from bacteria to human. This class of proteins is. And this might be just enough, just one mechanism. So it's like, for a physicist, it's an appealing idea that usually biology is quite messy, and here you just need one mechanism that make all work. What we also are now arguing is that during interface, when chromosomes are unfolded, and we have these fences and switches, loop extrusion may also play a very important role in bringing together the switches and their genes within a neighborhood. And if these loop extruders cannot go across fences, then it will guarantee that the switch only acts on these genes, and this switch acts only on those genes, and so on. So that's another idea that we just published a few months ago. So why is this all important? Again, it's kind of interesting and entertaining, but why is this important? First of all, my answer would be, first, it's general knowledge. The fact that sort of all museums have this stance, the fact that that Smithsonian Museum was running this human genome exhibition, tells us that this is something important. We would like to understand how genome is organized in 3D, and it may also be important for medicine. Again, speaking of general knowledge, so one of the first models from 2009 paper was picked for the cover of this new textbook in genetics. So it might also be, this chromosomal organization may also be important for medicine. For example, again, that's a paper published less than a year ago. There is a known condition with limb malformation in humans. Sorry for graphic images. What was not known and became known only recently is that it's controlled by one region of the genome in chromosome 2. And what was found in people with this disease is that what they're missing is just one fence. This single region where the fence is supposed to be is missing. As a result of this, you get interactions among two neighborhoods that were not supposed to interact, and a switch located in one neighborhood acts on a gene located in another neighborhood. And this leads to developmental mistake. That was a very, very important paper. It shows that all this fundamental knowledge that we accumulated over the last 5 years is actually important for medicine. Another example, cancer. Again, I told you that chromosomes are all deformed and carry lots of abnormalities in cancer. But again, a paper published also a few months ago shows that in glioma, in brain cancer, there is one particular region with a similar mechanism. A fence is modified. The region is not missing, but the fence is modified such that it loses its ability to insulate. And again, you get wrong interactions between a switch and a gene, and this leads to cancer progression of glioma. So it starts to look like that chromosome organization is important, but not only as a fundamental problem, but also is important for our understanding of Mendelian diseases, like this limb malformation and cancer. So let me summarize. So as I told you, genome is very long. It's about 6 feet long. Its folding is important for health and disease. We start getting some understanding of genome in 3D. These are the first steps. Again, we're roughly where structural biology of proteins and nucleic acids was in the early 1950s. We start getting first structures. The first protein structure was solved in 1956. The first DNA structure, practically the same year, 1952. Again, we start getting some ideas about the mechanisms. And chances are that this will change our understanding of how genes are regulated in general. So with this, I need to thank people who did all the heavy lifting, graduate students. In my group, these two have graduated. This was actually part of the work on this loop extrusion was done by a high school student, Carolyn Loop. She came to our lab as a high school student through MIT outreach program called Primes, and started working on this project, later got admitted to MIT, is now an MIT junior. Actually, these are pictures of students when they started this project. That's how they look now. I still use the same picture for Caroline because she didn't change. And our collaborators, they never change. Job Decker from UMass Medical Center, who developed high-C technology, and John Marker, physicist at Northwestern, who again has been developing this idea of loop extrusion. And again, obviously we're thankful to the funding agencies, NSF, Physics of Living Systems, and NIH recently started a new program called 4D Nucleome, trying to characterize nuclear organization in 4D, 3D plus time, and again funded several centers, including our Center for 3D Structure and Physics of the Genome. So we're very grateful for this funding, and thank you all for your attention.